There are various types of impurities in the drug substances and in the drug products, which may be organic impurities or related substances, which may be inorganic impurities like elemental impurities. Some are the toxic impurities like nitrosamines. Some are volatile materials or volatile impurities like residual solvents. Based on the impurity types, these are required to be controlled and these have specific limits. This video is regarding the enantiomeric impurities in the pharmaceuticals, and these impurities are the impurities which are closely related to the API structure. The impurities are required to be controlled so that the quality of the pharmaceuticals can be maintained. So in this video we are going to see what is the impact of enantiomeric impurities, how these are controlled in the drug substance and in the drug products, and what are the regulatory considerations. See, enantiomeric impurities are the enantiomers of the API or of the drug substance in the formulation. Enantiomers are molecules that are mirror images of each other and are not identical. So one thing we should know here, these impurities are the mirror images of the APIs. These are the impurities which are known to be coming from the synthesis of the API, or sometimes these may be coming from the degradation. Enantiomeric impurities refer to unwanted enantiomers present in the drug substance and intended to be a single enantiomer, and these may be active or inactive. Sometimes the drug substance is present in a racemic form. These enantiomeric impurities can impact the drug efficacy and safety. Enantiomers have different pharmacological effects, and the presence of an unwanted enantiomer may alter the therapeutic action or may lead to the side effects or adverse effect. Examples are like levofloxacin and levocetirizine. Levofloxacin, single enantiomer of ofloxacin. Levocetirizine is a single enantiomer of cetirizine. Now see the importance of chiral separation. Chiral, these are also known as chiral impurities. The chiral separation is essential in the pharmaceutical analysis, and regulatory guidelines mandate that only the active enantiomer should be marketed. Inactive enantiomers can act as a chiral impurities. That's why affecting the drug substance efficacy, safety, and bioavailability. Different analytical techniques and methods are used for their determination, like HPLC with chiral stationary phases, GC with chiral agents in the stationary phase, NMR, capillary electrophoresis, polarimeter, which measures optical rotation differences, X-ray, diffractometer, and differentiation by the melting point. These are some of the analytical methods for their determination. Now, how the enantiomers are named. Enantiomers are classified by the optical rotation, right-handed rotation and left-handed rotation. So R means right-handed rotation and S is the left-handed rotation. Naming follows the CIP rules, example dextrorotatory and reorotatory forms. So this is somewhat basic information about the enantiomers. Now see the regulatory aspects and ICH guidelines. ICH guidelines like ICH Q3A doesn't cover the enantiomeric impurities. The enantiomeric impurities fall under stereochemical purity instead of the impurity profiling. So what is the regulatory approach? These impurities are controlled through chiral synthesis and purification. Safety and efficacy evaluations are done to determine enantiomeric control. The regulatory authorities like FDA, EMA, and ICH require separate chiral purity assessment. And in conclusion, enantiomeric impurities must be strictly controlled due to their impact on the drug performance. Various analytical techniques can be used for proper identification and quantification. Regulatory guidelines focus on the stereochemical purity rather than impurity thresholds. Proper chiral separation is crucial for the pharmaceutical safety and efficacy. As per my work onto the products or APIs containing the enantiomeric impurities, these are controlled in the API mainly, and also these are controlled in the drug products. The enantiomeric impurities which are coming from the APIs have specific limit, and the same limits can be provided in the drug product. Sometimes some different limits are provided based on the nature of the impurities. Also, the monograph is required to be referred. Then, why the enantiomeric impurities are not covered by the ICH Q3A guidelines? Enantiomeric impurities are excluded from this guideline as they are not considered unintended process related or degradation impurities. But these impurities fall under stereochemical purity, which is handled separately. Actually, these enantiomeric impurities are generally the process related, but these are not the degradation impurities. This is observed that they are under the stereochemical purity type, and that's why these are handled separate. So, what are the key reasons for not covering the enantiomeric impurities in Q3A guideline? Enantiomeric impurities are not unintended chemical impurities. As the ICH guideline Q3A focus on organic, inorganic, and residual solvent impurities from synthesis or degradation. Enantiomers are not accidental contaminants, but these are the structurally related forms arising due to stereochemical synthesis. Then addressed separately under stereochemical control. Regulatory agencies expect enantiomeric purity to be controlled in drug substance specification. As I have uh, covered these under the ICH Q3A exclusion, these are controlled in the drug substance. So many of the times, if the enantiomeric impurities are not increasing in the drug product, you may address these uh, enantiomeric impurity in a separate way. Undesired enantiomers are managed through chiral synthesis, purification techniques, and analytical methods. Potential pharmacological and toxicological differences. So, one enantiomer may be active while the other may be inactive or toxic. Enantiomeric control is based on toxicological evaluation rather than general impurity qualification thresholds. This is very important to understand regarding the pharmacological and toxicological differences between the different enantiomers. Regulatory approach is different. The EMA, FDA, and ICH require separate chiral purity assessment for the chiral drugs. And instead of setting the impurity limits, manufacturers must justify safety, efficacy, and control of enantiomeric composition. Generally, the many of the drug substances control these impurities and they provide the specification limit in their COA. Sometimes these are controlled through the specification limits in the drug products in the similar way to the API or drug substance control. In conclusion, although enantiomeric impurities are organic impurities, they are excluded from the ICH-Q3A. They require different regulatory and scientific approach. These are controlled under stereochemical purity testing and specifications and not impurity profiling. That's why enantiomeric impurities have separate place in the COA of the drug substance and the drug product. But during the research phase, we need to evaluate these impurities whether these are getting increased on storage and stability or not. This is the basic information for enantiomeric impurities. If you have any queries, you can comment. I will try to answer your queries. But generally, as per my understanding and my experience, the enantiomeric impurities are limited in the drug products in the similar way to the APIs. Thank you for watching the video.